Hi everybody, I'm Har-Vardi. I'm the Director of Fertility and IVF Lab at Soroka University Medical Center and the Professor at Ben-Gurion University of the Negev in Israel. Welcome to Embryo Talk, where we are going to discuss cases from the IVF Lab and have a better understanding of the causes behind these cases. I'm very excited. Today we will talk about abnormal fertilization. After oocyte retrieval, insemination is performed using conventional IVF or ICSI, intracytoplasmic sperm injection. A fertilization check is performed 16 to 18 hours following insemination. As you are aware, normally fertilized oocyte consists of 2 pronuclei and 2 polar bodies. However, about 10% of inseminated oocyte are abnormally fertilized. If a zygote shows a single or more than 2 pronuclei, it is considered an abnormally fertilized oocyte. A 3 pronuclei or triploid zygote is classified as a D-andry when the extra set of chromosomes is from the father, and a D-gene when the extra set of chromosomes from maternal origin. Most cases of D-andry occur because of dyspermy, which means penetration of 2 sperm cells, and less commonly fertilization by 1 diploid sperm. Another option is duplication of the sperm chromosomes after penetration. On the other hand, most cases of the gene are caused by errors in the meiotic division during oogenesis. How can we distinguish between the two types of triploid fertilization? Firstly, it depends on how many polar bodies we see in the perivitellin space, and secondly, on the fertilization method, conventional IVF or ICSI. Basically, there are two different scenarios. The first one is 3 pronuclei with 2 polar bodies, and the other option is 2 pronuclei and only 1 polar body. But firstly, we need to understand what the difference is between the first and the second polar body. Let me explain. The first one is extruded after the elite surge or after an external trigger for ovulation. The second one is extruded after sperm penetration. Both polar bodies are of oocyte origin and consist of 23 chromosomes. However, the first one has 2 chromatids in each chromosome, and the second one consists of 1 chromatid in each chromosome. Let's go back to the 3 pronuclei scenarios. In the case of 3 pronuclei and 1 polar body, the extra third pronucleus has probably originated from the oocyte. The reason for this is incompletion of the oocyte's second meiotic division, and as a result of this, the third pronucleus reflects the polar body that didn't separated from the oocyte during the second meiotic division. When we see 3 pronuclei and 2 polar bodies, what is the source of the third pronuclei? In this case, the third one is originated from the sperm, since the second polar body is extruded after sperm penetration. This can be a result of penetration of 2 sperm cells, or alternatively, which is less common, a penetration of 1 diploid sperm cell. Cases of abnormal fertilization following ICSI mostly involve the formation of an additional female pronucleus owing to a non-extrusion of the second polar body, and consequently the development of the genic triploid zygote. You can see the attached video, which demonstrates a blastocyst development from 3 pronuclei zygote following ICSI. It is not a common case of 3 pronuclear fertilization, since the second polar body is extruded and then reabsorbed into the cytoplasm. It is difficult to follow the timings of the third pronucleus appearance and the polar body absorbance. In any case, the abnormal behavior of the second polar body is probably due to oocyte defects which caused abnormal fertilization. However, we cannot rule out penetration of an abnormal sperm cell. Some studies suggest that severe sperm abnormalities and oocyte aging can contribute to this process. In contrast, tripronuclear oocytes after conventional IVF are mostly caused by penetration of two sperm cells following normal second polar body extrusion. The transfers of embryos developed from tripronuclear zygotes can result in molar pregnancy. It is suggested that molar pregnancy develops due to an oocyte defect. This involves abnormal growth of the trophoblast cells that typically become the placenta. Molar pregnancies are defined as complete or partial. In cases of partial mole, there may be a fetus, but the fetus will not survive. In a complete molar pregnancy, there is only a placenta, but not a fetus tissue. Complete mole is usually diploid, but consists of only paternally derived chromosomes, since the oocyte does not express its genetic material. This results from the replication of a single haploid sperm after fertilization or fertilization by two sperm cells. The difficulty is that under the microscope we will see two pronuclei, such as normal fertilized oocyte. However, they are only of paternal origin. A molar pregnancy can have serious complications, including a rare form of cancer, and for that reason requires early treatment. Even though the embryos deriving from normally fertilized oocyte are capable of normal in vitro development, they are usually discarded because of a higher risk of miscarriage and of molar pregnancy. There is another option of tripronuclear zygote. This is known as 2.1 pronuclei, which means the appearance of two normal-sized pronuclei, but with additional very small one. The question remains, do we treat it as a tripronuclear zygote, or do we treat it like a normal fertilized oocyte? Do we transfer an embryo that is developed from such a zygote? In our next podcast, we will talk about this dilemma and try to answer these questions. Stay tuned.